Good.
You're good to go.
Okay. Hey everyone, greetings from the 7th floor conference room in the MBRB building. Just an open invite to anybody who would like to come and join for future seminars. We're going to do this for virtual talks. It's just been really fun to engage in science, at least from this one direction. So most welcome, and we do have snacks. So that said, it's my absolute honor and delight to welcome our speaker today, Heather McCauley. Heather is assistant professor in the Department of Cell Biology and Physiology here at UNC. As a grad student at University of Cincinnati, Heather used conditional tissue-specific knockout mouse models and discovered a new role for TGF-beta in goblet cell differentiation in the ocular surface epithelium. Interestingly, she found that targeting TGF-beta signaling can help treat disorders such as dry eye, which is something that I myself suffer from. So thanks, Heather, for doing good work. She went on to postdoc with Jim Wells at Cincinnati Children's and was quickly promoted to instructor. Heather's used organoids to demonstrate that the neuropeptide YY is a key regulator of macronutrient absorption in the small intestine. She's also discovered that enteroendocrine cells are essential regulators of intestinal metabolism. So we're really in for some exciting work today. Heather started her lab at UNC just in November. She's barely been here a year. So everybody, please join me in welcoming Heather to the Carolina community as well as the CGIBD. So Heather, take it away.
Thank you, Audra. And thank you for being my friend over the last year and helping to introduce me to all of the CGIBD community. I'm really happy to be here. So today I'm going to tell you about enteroendocrine cells, and you probably know a little bit about them, but we'll start from the beginning anyway. I am really motivated by this overall question of how does the food we eat change our physiology? And in response to a meal, the body goes through many different physiological processes, such as signaling satiety to the brain, triggering a digestive enzyme release from the liver and gallbladder, and glucose regulation from the pancreas. And all of these whole body responses to food are made possible by enteroendocrine cells. And so therefore, enteroendocrine cells are really the primary link between the food we eat and the signals received by the rest of the body. However, much less is known about how enteroendocrine cells regulate their local environment within the intestine, which is really the focus of my lab. But first, we need to understand more about enteroendocrine cells for a number of GI diseases that impact a large population globally. So everyone who eats food should have a well-functioning enteroendocrine cell system, as these cells are really dealing with every physiological response after you ingest a meal. Enteroendocrine cells are now therapeutic targets for type 2 diabetes and obesity. If you've heard of Ozempic, and I don't know how you haven't heard of it at this point, that's targeting one of the enteroendocrine cell hormones that signals to the pancreas to regulate glucose control. Enteroendocrine cells are frequently altered or dysfunctional in people with obesity and those who suffer from inflammatory bowel disease, which collectively affects a large segment of the global population. And I'll show you some data today that suggests enteroendocrine cells might be potential therapeutic targets for the treatment of malabsorption due to diarrhea, which is a leading cause of childhood death. And there's about 2.5 billion cases in children under 5 per year. And so understanding enteroendocrine cells is really an important foundational step to understanding human physiology. However, like I mentioned, comparatively little is known about how enteroendocrine cells regulate their local environment within the intestine itself. And so that's really the vision for my new group that's here at UNC. So enteroendocrine cells are a rare population of cells. They only make up about 1% of the intestinal epithelium, but they sense environmental stimuli that are passing through the gut, like nutrients, microbes, and microbial metabolites. And in response to these signals, they secrete about 20 different hormones that go into the bloodstream and affect those systemic organs, collectively forming the largest endocrine organ in the body. But we knew enteroendocrine cells were important for local function within the intestine because of this phenotype where a few patients, very rare, were reported that don't have any enteroendocrine cells and they have malabsorptive diarrhea, indicating that these additional cells within the intestine are being targeted by enteroendocrine cells. So these patients were all babies that were born at a normal birth weight. There was really no indication there would be anything wrong with them. But within a few days after birth, they all came back to the hospital with this intractable malabsorptive diarrhea upon ingestion of any oral nutrients. And all these babies needed TPN in order to gain enough weight to survive. So they sequenced these patients and found they all had mutations in the transcription factor neurogenin-3, which we had known from animal studies to be required for enteroendocrine cell formation. And sure enough, when they did biopsies of these kids' intestines, they looked mostly normal, with the notable lack of these rare enteroendocrine cells, which are shown here in brown in a normal patient. But this really was puzzling to the physicians. Why loss of this one rare cell type would impact the function of the remainder of the intestine, especially the absorptive enterocytes, which are shown here in yellow. And so this was the question that really drove my curiosity when I started my postdoc in Jim Wells's lab at Cincinnati Children's. And we decided to approach this using human intestinal organoids. There are two different types of organoids. I think most people here are more familiar with organoids that come from a surgical biopsy specimen. You can harvest the intestinal crypts from there that have the tissue-resident adult stem cells, put those in the right culture conditions in vitro, and grow these epithelial-only organoids, or enteroids. There's another way to make three-dimensional human intestine in a dish, and that's by starting with pluripotent stem cells, which can either be induced from fibroblasts, skin, blood, or come from the inner cell mass of a fertilized blastocyst. These are called embryonic stem cells. And we really haven't made any of these since the 90s because they perpetuate and they self-renew like crazy. So these two ways are the sources for pluripotent stem cells, which can become any tissue in the body when given the right signals. And so we direct their differentiation into intestinal tissue by giving these cells the same growth factors and signaling pathways that they would experience during embryonic development. And so these grow these spontaneous little three-dimensional spheroids, which when you plate them in 3D, grow into these nice intestinal organoids that have not only the epithelial layer, but also the mesenchymal layer, which will go on to make fibroblasts and smooth muscle and some vasculature and all of the other supporting cells that help the epithelium function. In collaboration with the Helmrath lab, who actually started his program here at UNC several years ago, we developed a technique to transplant these intestinal organoids into the kidney capsule of a recipient mouse. If you let them grow for about 2 to 3 months, they mature into this beautiful crypt villus architecture, which really looks just like a biopsy from a baby's human intestine, with this nice crypt villus architecture. All of the differentiated cell types are there. And what this allowed us to do is use these transplanted organoids as our own biopsy source, where we can harvest the crypts from these organoids and put them in culture and make these enteroid structures without needing any surgical biopsies. And this was really important for these kids that have the mutations in neurogenin-3 because they're very rare, they're very sick, and we were unable to get any biopsy specimens from these kids. So we used CRISPR gene editing to introduce a mutation in neurogenin-3 and directed their differentiation into intestinal organoids and drove them through this process. And we result in human intestinal epithelial tissue that lacks enteroendocrine cells, which are shown here in green in a wild type sample. And so we're using this model, human intestinal organoids with and without enteroendocrine cells, as well as mouse models to find new roles for enteroendocrine cells. And today I'm going to tell you about two projects that I directed during my postdoc that are really the foundation of my new research program here at UNC. And then I'll end on just one of the things that we're working on in the lab now. So I was really motivated by this question of how enteroendocrine cells are regulating the absorptive function of their epithelial neighbors. And so we went back to the literature and learned about nutrient absorption. And we've known for probably 100 years at this point that glucose is absorbed coupled with 2 sodium ions, and di and tripeptides are absorbed coupled with a hydrogen ion. And the absorption of these nutrients is really dependent on the ion gradients across the apical membrane. And that's established and supported by a number of ion transporters across both the apical and basolateral membranes. But for today, we're just going to focus on 2. This is CFTR. It extrudes chloride from the cell. And this is NHE3, or sodium hydrogen exchanger 3. And these are really important for maintaining these gradients across that support nutrient absorption. And while we've known how this process works for a long time, there was really no indication that anything would be regulating this process other than just nutrients passing through and the cells are capable of absorbing them, so they absorb them. But these patients that don't have enteroendocrine cells gave us a clue that enteroendocrine cells might be an important player in this process. And so we found an example of this in the colon, where one of the hormones made by enteroendocrine cells, called PYY, actually balances a neurotransmitter made by enteric neurons called VIP. And these exert opposite effects on the activation of cyclic AMP within the cell. And cyclic AMP will drive the opening of CFTR and chloride extrusion while inhibiting NHE3 function and the exchange of these ions. And so we hypothesized that if this mechanism was also acting in the small intestine, this might be upstream of setting these gradients that are required to regulate ion-coupled nutrient absorption. And so we predicted in the patients without enteroendocrine cells, they wouldn't make any PYY, but they still have enteric neurons. They're still making VIP, which is this signal that leads to the accumulation of cyclic AMP. Excess cyclic AMP leads to a lot of chloride being secreted from the cells, and sodium and water will follow. There's an inhibition of this sodium hydrogen exchanger, furthering the imbalance of sodium and hydrogen across the apical membrane. And we expected that this would establish these gradients that are not supportive of normal nutrient absorption, where we'd expect glucose to actually enter the cell at an accelerated rate and di and tripeptides to not really be absorbed at all due to the intracellular pH. And so we sought to test this hypothesis using our enteroendocrine cell deficient models, as well as use these models to find candidate hormones which could impact nutrient absorption. And this is important not only for these few kids that have this rare mutation, but we thought this might establish a new mechanism for the treatment of diarrhea and malabsorption, which, as I mentioned earlier, was a major cause of death in children worldwide. And the workhorse for these experiments is the Using chamber, which is common in the lung field and less common in the GI field these days. But you essentially create a battery where you can mount a piece of epithelium in this chamber that separates media on the basolateral side from the apical side. And there's electrodes connected to this Using chamber, which means you can measure the electrical resistance, the voltage, the current that's passing through this epithelium in response to anything that you expect to elicit an electrogenic response. And so we were able to add all of our candidate factors to the Using chamber in both mouse small intestine and human intestinal organoids with and without enteroendocrine cells. And long story short, we found that our model was, we did predict correctly with our model. So we find an increased response to VIP in enteroendocrine cell deficient tissues compared to normal, which could be normalized by the exogenous addition of PYY to these tissues that don't make any enteroendocrine cells of their own. This also extended to regulating glucose absorption, Because glucose is absorbed coupled with sodium ions, we can measure that as a function of electrophysiology. And we find a faster rate of import in our enteroendocrine cell deficient tissues compared to normal. And this is significant because the patients have this severe diarrheal response to glucose, which is accelerated by this downward gradient. We found that addition of PYY could restore the slope of that curve back to normal, indicating that PYY is actually sufficient to regulate glucose absorption in the small intestine. We were really curious to see whether we could use this as a preclinical model to test the therapeutic ability of PYY in enteroendocrine cell deficient patients. And so we used the Villain-Cree neurogenin 3 floxed mice. These mice were previously made by Gerard Gradwell's group in France, and they are very sick. They recapitulate the phenotype of the human patients. They have no enteroendocrine cells from before birth. They have horrible malabsorptive diarrhea. About half of them will die within the first week of life, and most of the rest of them will die surrounding the weaning period. We decided to start treating these mice that are still surviving at postnatal day 10 with once daily injections of PYY. And we found we could improve their diarrhea and greatly improve their survival across the weaning period. These mice they gained a little bit of weight. They had a normal electrophysiological response to VIP and glucose in the oosting chamber. But we were really curious to see about protein. So from our model, we predicted that this gradient would not support dipeptide absorption. And in fact, that's what we saw experimentally using the oosting chamber. There's really a flat line in response to this dipeptide. This is important because they did a follow-through study in the index patient and recovered about 80% of the ingested protein in his stool, suggesting that protein malabsorption is a particular problem for these kids. We found in our rescued tissues that had been treated long-term with PYY, a restored normal response to this dipeptide, again, indicating that PYY is sufficient to regulate protein absorption in the small intestine. So we screened a couple of hormones for which ones would influence this process, and PYY was the best. Everyone asks what the other hormones are. Somatostatin does the same thing, but we found a much greater response to PYY. And while total loss of enteroendocrine cells is rare, poor absorption is prevalent from things like enteric pathogen infection, Crohn's disease, and short gut or intestinal failure. So we really think that this is a mechanism that could be exploited for the treatment of many different diseases. But for me, as a scientist, what was really exciting about this project is it opened the door to see how else enteroendocrine cells might be regulating their neighbors in a local fashion. And because we observed these changes in ion stores, not only ion gradients across the membranes, we questioned whether there'd be differences in intracellular ion stores, which are intimately linked to cellular metabolism and mitochondrial function. So our next hypothesis was that enteroendocrine cells are regulating cell metabolism in the gut. Most people don't know this, but the intestine is actually a major metabolic organ, consuming about 25% of whole body oxygen uptake, which fuels mitochondrial respiration. And so we predicted that factors which influenced intestinal metabolism would have exponential effects on systemic metabolism. We all know there's two major compartments of the small intestine. We have crypts and villi. The villi are made up of majority enterocytes, which are chock full of mitochondria, which support the active transport of ions and nutrients, like we just discussed, as well as all of the processes of lipid trafficking. Conversely, in the crypt, this is where the stem cells live. It's a highly proliferative compartment, and they run on an entirely different metabolic program. Where mitochondria still are essential for their normal function, but comparatively to enterocytes in the villus, there's very few active mitochondria in the crypt. And because enteroendocrine cells are found in both compartments, we hypothesized that they might be regulating this switch, this metabolic switch and accumulation of active mitochondria in the villus. And so first we tested whether enteroendocrine cells could regulate whole body metabolism by putting animals in metabolic cages. And you may notice now we're switching to an adult inducible model of loss of enteroendocrine cells in the intestine. This is for a couple of reasons. One, we did not want to analyze any secondary effects due to lifelong malabsorptive diarrhea. And two, most of the animals with the constitutive deletion of neurogenin 3 die before crypts and Paneth cells are really entirely mature around P14 in neonates. So by using adult animals, we can circumvent both of those problems and really study what happens to the whole animal and the intestine with an acute loss of enteroendocrine cells. And interestingly, we found that immediately upon loss, the first tamoxifen injection, loss of enteroendocrine cells, we see an increased total energy expenditure in our enteroendocrine cell deficient animals compared to control. And this equation is essentially a measure of how much oxygen the animal is breathing in and how much carbon dioxide they're breathing out. And so we see an increase in this energy expenditure in the absence of any changes in food intake or activity. And so this was great. This suggested that our hypothesis we might be on the right track, that changes in intestinal metabolism might be affecting systemic metabolism in a big way. So let's look at the intestine. We harvested crypts and villi from our wild type and our enteroendocrine cell deficient animals and performed NMR metabolomics. And just like has been predicted in the literature, we found a nice separation in metabolic profiles between cells isolated in the villus and cells isolated from the crypt. However, when we look at our enteroendocrine cell deficient tissues, we lose that separation of metabolic profiles. And when you place all 4 of these samples on the same PCA plot, you can see that knockout crypts occupy this space in between control crypts and villus samples. And because mitochondria are really the major, is really one of the major players in establishing this difference in metabolic profiles, we decided to focus on how enteroendocrine cells might be regulating mitochondrial activity in the intestine, especially in the crypt. And so to do these experiments, we turned to a live imaging approach where we use this dye called TMRM. It labels active mitochondria where the fluorescence intensity correlates with the potential of the inner mitochondrial membrane. So the brighter the signal is, the more active those mitochondria are. And just because there's no signal doesn't mean that there's no mitochondria. It just means that relatively speaking, they're not nearly as active as the ones that take up a lot of this dye. And so again, we separated our crypts from villus. And just as predicted by the NMR metabolomics, we see active mitochondria in villus cells from both control and enteroendocrine cell deficient animals. However, when we looked at crypts from these animals, we found a striking difference where there's very few active mitochondria in wild type crypts and abundant active mitochondria in crypts from enteroendocrine cell deficient animals. And you can see here at this exposure level, which is like the laser is barely on to get this, this amount of fluorescence intensity. You can barely see, there's a few small dots where there's active mitochondria in the control samples, but compared to the knockout samples, there's a dramatic difference in mitochondrial activity. And so we took a step back and wondered, what does this mean? So loss of enteroendocrine cells is really loss of the nutrient sensing cell. And so we questioned whether loss of nutrient sensation would mimic what happens when cells are deprived of nutrients. And there's been several studies on what happens to the crypt and intestinal metabolism in fasting models and different dietary challenges. And so we found when we fasted control animals overnight, we also saw this accumulation of active mitochondria in the crypts upon a 16-hour fast. We then wanted to determine whether this active mitochondria correlated to any functional changes. And so we used a seahorse assay to demonstrate that these enteroendocrine cell deficient and fasted crypts are actually running on increased fatty acid oxidation, which is a major function of the mitochondria. And so this is going to be complicated, so I'll try to make it as simple as possible. at baseline, we can record the oxygen consumption of crypt cells. And that's basically how much oxygen is coming into the mitochondria for oxidative phosphorylation. And we find at baseline that enteroendocrine cell deficient and fasted crypts have a much higher oxygen consumption rate than control crypts. So this suggests that those active mitochondria we were seeing are actually consuming oxygen and doing something functionally. We then tested whether it was fatty acid oxidation by blocking this CPT1A, which is the essential transporter to bring fatty acids into the mitochondria for fatty acid oxidation. And this drug, etomoxir, specifically blocks this transporter. And so when we add etomoxir, we see two important things. One is that we've now reduced that increase in active mitochondria in both our knockout and fasted samples. So now these are not distinct from wild type crypts when you block this fatty acid oxidation. And secondly, when you use etomoxir to block fatty acid oxidation in control crypts, you see no difference in the oxygen consumption rate, indicating that in a wild type control situation, these crypts are not relying on fatty acid oxidation as a major metabolic program at all. We further wanted to test this by looking at transcription of lipid metabolism genes. And so we did a single cell RNA sequencing of our transplanted organoids, human organoids, with and without enteroendocrine cells. And we focused just on cells that live in the crypt, so stem cells, progenitor cells, and Paneth cells. And when we ran the gene ontology for these three populations, we find all 20 of the top Go terms are associated with metabolism or metabolic processes in stem and progenitor cells. And a large number of them are also popping up in Paneth cells, which is very strange because Paneth cells have very few mitochondria and they're almost entirely glycolytic. But this suggests that enteroendocrine cells are important regulators of lipid metabolism within the crypt. And so we next wanted to know functionally what this means. And so several groups have shown that intestinal stem cell activity can adapt to changes in cellular metabolism within the niche. And crypt metabolism and intestinal stem cell function are really responsive to changes in diet, like fasting, caloric restriction, or high-fat diets. And so we wanted to test intestinal stem cell function. So we did this in a couple of ways. First, we looked at proliferation, and we find a slight but significant increase in both Ki67 and EDU incorporation in our enteroendocrine cell deficient crypts, which is equivalent to the levels you see after an overnight fast in control animals. We also saw this in our human intestinal organoids, where there's a small but significant increase in proliferation in the absence of enteroendocrine cells. But the gold standard of testing intestinal stem cell activity is the enteroid formation assay. So we all know that from mice, you can harvest intestinal crypts, put them in the right culture conditions in vitro, and they make intestinal enteroids. And this is a direct reflection of the ability of intestinal stem cells to self-renew and differentiate. We can do the same thing with our human intestinal organoids by harvesting the crypts and putting them back in culture in the right media conditions. And by using these assays, we found that loss of enteroendocrine cells actually increases stem cell activity, where you have greater enteroid forming capacity compared to control. And again, this is equivalent to a fasting model. The knockout enteroids grew much faster and needed to be split more frequently over the first couple of passages. But then over time, this difference normalized, which we think is due to loss of endogenous signals in a physiological model and normalization of these cells to the really high Wnt conditions in the growth media for the enteroids. And so in trying to think about a mechanism of how this might be happening, I was reminded of this paper, which I loved when it came out 10 years ago. It was such a cool paper from Omar Yilmaz from Omar Yilmaz when he was a postdoc, and he's continued on this work in his own lab, where Omar and his group found that when mice are fasted overnight, they lose active mTOR signaling in the crypt, especially in the Paneth cells. So normally in a control mouse that's eating chow ad lib, you see really nice active mTOR signaling indicated by phospho-S6 in the crypt and especially in the Paneth cells, so co-localizing with this purple marker. But when mice are fasted overnight, you lose all of that mTOR activation in the crypt. And mTOR is really this master nutrient sensing pathway that has a number of downstream effects, telling the cell whether you have enough nutrients to perform all these duties or you don't. And so we recapitulated Omar's findings in our own hands. These are from my colony. And interestingly, we found, just as we had shown from the other experiments, that loss of nutrients really is the same as loss of the nutrient sensing cell, where now these mice have no enteroendocrine cells, but they're eating plenty of chow. They have plenty of nutrients floating around, but that signal is not making it down to the crypt. So the crypt thinks that it's fasting, even though there's plenty of nutrients. And this also was observed in our enteroendocrine cell deficient human intestinal organoids. And so this leaves us with a model where enteroendocrine cells are really these essential relay cells, where they sense the nutrients that are passing through the gut and in turn transmit that signal to cells deep in the crypt that don't really have access to luminal nutrients by regulating mTOR signaling in the Paneth cells and controlling the level of fatty acid oxidation, proliferation, and intestinal stem cell activity. So these are all the things I just told you, that enteroendocrine cells are really protecting the stem cell niche and instructing them on the nutrient status of the gut. And so these two projects were complete. I completed them when I was a postdoc, but they really form the basis of what I hope to continue in my own lab here at UNC. And importantly, we have to think that the intestine is not just epithelial cells. There's a number of other cell types that are essential for normal intestinal function, absorption of nutrients, whole body health and physiology. And so I'm expanding the scope of my research program to look at how enteroendocrine cells are regulating the local cell biology and function of their neighbors in response to nutrients and microbes in stromal populations like immune cells and the enteric nervous system, as well as in diseases like inflammatory bowel disease and cancer. And so I'll end just on a little snippet of one of the projects that we've started now, which really ties back into cancer. And so from the work I just showed you, we found that enteroendocrine cells restrict proliferation and intestinal stem cell activity. That suggests they might be acting as tumor suppressors and regulating the amount of proliferation that cells are able to undergo. In conjunction with that, are they cooperating with classical signaling pathways like Wnt and BMP, which are known to regulate cell proliferation? And so I wrote up a grant to study this, and I'm very happy to say that this project is being supported by the pilot and feasibility from the CGIBD, where we're looking at really these two questions. Are enteroendocrine cells regulating, are they influenced or influencing the Wnt and BMP gradients across the crypt-villus axis? And are they participating in tumorigenesis in a model of adenoma that has high Wnt signaling? And so first, we found that in the absence of enteroendocrine cells, crypts from mice actually display this increased nuclear phosphosmad signaling, which is the downstream effector of BMP. And normally along the crypt-villus axis, there's very low BMP in the crypt, and it increases as you move up the villus. And this is due to the presence of BMP inhibitors and antagonists that are made by stromal cells down in the crypt. And so we hypothesized that enteroendocrine cells might be acting on these stromal cells, which are regulating the amount of BMP inhibitors that are in turn acting on the intestinal stem cell niche in the crypt. And so to address this, we're doing a couple of things. We're doing a lot of RNA-scope and immunofluorescence to really nail down what cells are producing what signals and how enteroendocrine cells are influencing that. And we are gearing up to do a nice big single cell sequencing experiment with Scott's help to look at not only the epithelial cells, which has been done before, but the mesenchymal cells in the intestine with and without enteroendocrine cells. And secondly, we wanted to investigate whether enteroendocrine cells are participating in this Wnt-mediated tumorigenesis, such as an APC-Min model, where constitutively active Wnt signaling leads to abundant abnormal proliferation, causing these adenomas and putting the tissue at risk for more mutations that would really drive colorectal cancer. And so we're doing a number of things in this project as well. Notably, we crossed our enteroendocrine cell deficient mice to the APC-Min model, and we're in the process of looking at our first pilot group of animals to see what's going on with them. And we'll further do some sequencing and some positional analysis of what cells are doing what. But I'll show you a sneak peek of the data. So in APC-Min animals, they develop these spontaneous neoplasias starting at about 4 months of life. So we decided to analyze our mice at 4 months, both with and without enteroendocrine cells. So this is a pretty early time point. And we found that without enteroendocrine cells, we see a large number of these intestinal adenomas in the small intestine compared to APC-Min mice that still have their full complement of enteroendocrine cells. And we don't have the power to do statistics yet or to find significance yet. But we found that most of our enteroendocrine cell deficient animals have this huge number of tumors across the small intestine compared to control animals. And so I'm excited to make some progress on this. I think this is a pretty unique project in both looking at stromal contributions to establishing the Wnt and BMP signaling gradients, as well as enteroendocrine cell influence on tumorigenesis, which I think is very cool. So I'm really excited to pursue this project and others with the help of my new team. All of these people were brave enough to join a brand new lab, and I'm really excited to work with them. Everyone's extremely talented and motivated and excited. So yeah, that's my new lab, my new ideas, and happy to talk about anything you want.
That was terrific, Heather. Thank you for that. As your host, I'm going to ask the first couple of questions, if that's okay. But for everyone else, either please put your hand up or unmute yourself and you can ask the questions. So Heather, can you talk a little bit about what happens to EEC sensing when the mucus layer is eroded, such as in the case with starvation, as has been reported? And then sort of related to that, how do you think that microbiota contribute to PYY synthesis specifically?
Mm-hmm. Yeah, both really good questions. Me personally, I have not done anything with mucus or the microbiome yet, but that's one of the reasons I came to UNC, was to take advantage of the wonderful Notobiotic core and kind of look at this. So we expect that when there's a thinner mucus layer, that more of the luminal cues, the metabolites and whatever, are going to be sensed by the epithelium. But conversely, in conditions like IBD, where the mucus layer is disrupted, you actually see a decrease in enteroendocrine cells and hormone secretion. So there's still a lot to kind of unpack there and see what is driving what phenotype and how enteroendocrine cells, whether they're upstream of anything or they're just responding to a change that's driven by another process.
I have a question.
Yes, go ahead.
Oh, thanks, Audra. Heather, that was awesome. This is odd, by the way, sir. My question is, how do inflammatory cytokines like TNF or TSLP or IL-8 within the intestine impact EEC, viability and function? And are there direct immune cell interactions with an EEC in the gut that are notable that we know of?
So the most work has been done with enterochromaffin cells, which make serotonin. That's one of the subtypes of enteroendocrine cells. And they actually can directly recruit macrophages and T cells, I think, within inflamed regions of the gut. They have the receptors on the cells, and they've done in vitro experiments where they just put serotonin in and it causes an inflammatory, like a cytokine release from these cells. In terms of how the immune cells and their cytokines influence the enteroendocrine cells, like from the inside, I don't think anyone has studied that. What we do know is that there are TLRs and other immune-mediated sensors on the apical surface that can measure the microbes and the metabolites. But I don't think anyone has looked at the bottom side up and what these cytokines and inflammatory cells are doing once that inflammation has already started.
Thank you.
Tyler, do you want to unmute yourself and ask your question?
Sure. So I'm Tyler Culpepper. I work in Janelle Arthur's lab. And my question is really about the distribution and the function of these enteroendocrine cells in the small versus the large intestine and how that is associated with cancer. And you showed the APC-Min model, which is predominantly small intestinal tumors. And a lot of this nutrient absorption data comes from small intestinal applications. But most of the cancer we see in humans are, intestinal cancers, colon cancers. So are there notable differences in the physiology or the distribution of the enteroendocrine cells that would make it a little bit more difficult to apply, or is the model representative of both?
Yeah, that's something I think about all the time. So there are gradients of hormones produced by enteroendocrine cells from proximal to distal. And many people in the field even have incorrectly assumed that certain cell types are only found in the small intestine and certain are only found in the colon. But once we've done all of this transcriptional profiling, we find that these classical subtypes are actually, it's an oversimplification, that a lot of these cells can make the transcripts for 8 or 10 hormones, and depending on their proximal to distal location, that's the ones that they actually secrete. So you have more GIP and CCK in the small intestine and more PYY and GLP-1 in the distal small intestine and colon. But you can find all of the hormones across the whole axis. So it's just a matter of ratio, first of all. I have thought a lot about how to put this in the colon, to relate it to colon cancer. What I think is that the principles of how enteroendocrine cell hormones are interacting with the Wnt pathway might be the kind of like mechanistic way that we're going about this. And hopefully we find something there that is also happening in the colon, which we can't really test in the APC model, but we could test with human samples or something like that. So I'm pretty much looking for a something testable in the small intestine.
Well, I think it's already a great model for familial adenomatous polyposis, FAP, which is basically, I'm sure is the human mutation. I had one more minutia question, if I can, since we have a little time. I was just curious. I'm going to start working with BirdU as well and looking for proliferation. And I saw your IF there. And I saw you had KI67 as well. do you need to counter stain to actually assess that? Or can you just use just the Bordeaux?
A lot of people just use one or the other.
Okay.
It's my understanding that Ki-67 marks multiple phases of the cell cycle, whereas BRDU or EDU incorporation is only tagging cells in S phase. So you can get a little bit of a different picture of what's happening with the cell cycle dynamics by using both, which I haven't done, but I have the data if I ever needed to look at it.
Okay, appreciate it. That's all I had. Thank you.
Any other questions from the Zoom audience?
I have one question. That was a great talk there. I'm wondering because, especially work from Duke has shown that the enteroendocrine cells will be so involved with innervation of the intestinal epithelium. Do you think that that could have to do with any of these phenotypes, or do you think that like the wind, the hormones, and the innervation might all just be kind of doing separate things?
I think everything is involved. I think it's nutrients and microbiome and immune cells and enteric neurons. And my kind of central idea is that enteroendocrine cells are kind of coordinating all of these different interactions with all these different cell types, keeping everything running. Yeah, so the first project I talked about is looking at what happens when you have enteric neurons, but you don't have enteroendocrine cells. So it's really kind of breaking that connection of, or showing that enteroendocrine cells are required to balance signals from the enteric neurons.
Sure.
A lot of hormone receptors are expressed on enteric neurons, more than there are on the epithelium itself. So for example, GLP-2, which can increase the intestinal mass in people, like it's being used for short gut syndrome. The GLP-2 receptor is not on the epithelium. It's actually on enteric neurons and I think endothelial cells. And so it's really a secondary process by which enteroendocrine cells are regulating that process. And I think that's going to be the case for a lot of things.
Cool. Thank you. And great work.
That's great. Thank you. Well, if there's no one else, everybody, please join me in thanking Heather for a great seminar, and see you all next week. Thank you, Heather.
Thanks, Heather.